20 years ago, a diagnosis of multiple myeloma meant 3 to 5 years to live. Today, patients are living for decades with CAR T, bispecifics, quad therapies.
The prediction now after a quadruplet transplant is that these patients are probably going to go over 10 years on average before their first remission.
Moving from trials into practice, patients are finally gaining real access to therapies unthinkable just a few years ago. This field is dynamic. There's a lot to choose from.
We have 20 drugs if you take the whole field over the last 2.5 decades, and in that we need all of them.
We're closer than ever to transforming myeloma into a chronic disease and maybe even asking, can we cure it?
We have an opportunity to potentially bring in more functional cure therapies for patients.
I'm Dr. Tania Small, and on this episode of Doctors Unscripted, I'm joined by 3 remarkable voices. Dr. Paul Richardson of Dana-Farber and Harvard, who has helped pioneer nearly every major therapy in use today. Dr. Joseph Mikhael, Chief Medical Officer of International Myeloma Foundation, a true change agent who has shaped how we think about myeloma. And Dr. Mecide Gharibo, who went from Associate Professor of Hematology and bone marrow transplant to driving the development and delivery of new medicines in hematology. Together, we'll cut through the complexity and ask the provocative questions, shaping the future of myeloma care.
Now let's get started.
Before we go into the science, I want the listeners to get to know you. So if you could do me a favor and think back, way back, to when you were in med school. Were you able, you were able to identify that person who was going to go into plastics, or that person who was going to go into ortho, and definitely you could figure out who was going to go into psych. Did your colleagues know, what did your colleagues think you were going to go into, and what made you decide to go into hematology? And I'll start with you, Mecide.
Well, I knew at very early age that I had a passion for going into medicine, but more importantly, hematology was something that I was very much interested in. So through my undergrad, I had an opportunity to spend some time in research labs, and then going into medical school, I actually did my best to get as many electives as possible.
In heme?
In inpatient leukemia service, outpatient hematology clinic at different hospitals that we had an opportunity to rotate through. So my classmates knew that my passion was heme malignancies, and I loved the fact that the complexity of the spectrum of heme malignancies, but more importantly, that we had made so much advancements that we could actually talk about a cure for patients with leukemia.
Yes.
And then while I was in medical school, I saw the advancement in multiple myeloma. That's when things were starting to emerge with bortezomib, and then you had lenalidomide emerging in the treatment. So we started to see the impact of those therapies. So it really excited me to be able to go into a field.
Yes.
And just build on where that field started.
What about you, Joe?
I think if you ask my classmates in med school, they would say that I would go into internal medicine because they all knew that at some point I wanted to be involved with international health, and that was going to maybe facilitate that. But then really what ultimately happened, to what I call the double hit of going into what I did, was my mentors, right? We know that actually most medical students are very heavily influenced by who they had as mentors. And the individual who was assigned to me to teach me physical exam skills was a hematologist. And to this day, I'm grateful for him. And then lastly, when I was an internal medicine resident, I was called by the emergency doctor to help him with a case that didn't make sense. It was this young guy. Remember, I grew up in Canada. He had been playing hockey. He got checked into the boards, and they showed me his x-ray, and he had all of these fractures and all of these lytic lesions in his spine. Turned out to be one of my friends from high school.
Oh.
He's still with us today,
Wow.
almost 30 years later, which is a miracle. He was one of the few people that had myeloma and had an allo transplant from his bigger brother. And that made me realize I really wanted to take care of the sickest of the sick.
Yes.
And those were days where sadly myeloma survival was maybe 1 to 2 years. And here we are 30 years later with survival, as you know, well over a decade. So,
yeah.
And I don't regret a single decision because I love what I do.
So what about you, Paul?
Well, my mother was a nursing director, so my early life was literally with her in the hospital, because I'm very sadly, my father passed when I was very young, so my mum was basically single, a widow, and so she would take me to the floors. So my first experience of medicine was serving sort of tea and biscuits to patients for my mum, and then it was actually great. And then I was very fortunate in the high school to spend time with a really gifted biology teacher who gave me a project in cancer. And basically I got absolutely intrigued in it, and just because of his good guidance, it got a prize. And so basically I thought, this is amazing, what is cancer all about? And then went to a fabulous med school, St Bartholomew's in London, Barts, and it, by happenstance, had a very strong tradition in cancer medicine. And I had the privilege of working, just as you alluded to, Joe, with some mentors who really could just drive that. And I'll never forget my yearbook. At the end of my yearbook, one of my buddies wrote the yearbook, and he had a picture of me, and he said, "Sorry guys, I'm late, but all of my mates have got cancer." And it was just, it was so funny, because what he was doing was taking a big swipe at me, because I tend to run a little behind.
I just heard just a little bit.
Yeah. And then, so,
What? I've never
Just a
heard that,
little
Paul.
bit.
I think my mum explained that I was a few weeks overdue, so I never caught up. I had the privilege of working with some really great people, and one was Jim Malpass, who was a founder in myeloma. And I'll never forget my first myeloma patient with Jim. She was a lovely matriarch from East London, and I admitted her, and 3 months later, very sadly, had to sign her death certificate. That was back in 1986, and I'll never forget her. I used to take her down to the square at Barts so she could look at the sky, because she was so devastated by bone disease. She was bedridden, and so to give her time to actually see daylight and sunshine, and it was just such a moving experience with her. And then I also had the opportunity to work with Tim McElwain, who was the founder of high-dose melphalan. And it was amazing to work with Tim, because what we would do is we'd do autologous stem cell transplant, and then we would use alpha interferon as immune therapy. The problem was the toxicities. And I'll never forget Tim McElwain saying to me, and I was stunned, he looked at me and said, "Paul, if we can do better than high-dose melphalan, we'll be doing our patients a service." Wow. And here we are today.
Yes.
So, it's very interesting, and it was a privilege to have that background, and so that's kind of what took me in that direction.
Very interesting. And it's interesting, I guess, except for you, most people knew they were going to go into, because I can tell you, I actually thought I was going to go into OB initially, and then I ended up I did a rotation, actually in med school, with my old mentor, who they used to call a cowboy, because we were transplanting sickle cell patients. Wow. And curing them.
Yeah.
And so I rotated with him in med school, and then ended up going to do my fellowship under him, and that's what brought me in. So it's actually the sickle cell route. So benign heme brought me in to then heme, and then in peds, because I was peds hemonc. You do heme, solid, as well as transplant all in one. As a fellow. So that's what actually brought me in.
Wow.
I want to talk about the biology of multiple myeloma, because if you think about even years ago, treatment has changed dramatically. Before we just had a few options. Now the field is dynamic. There's a lot to choose from, and there's a lot that we've learned both from the biology standpoint as well as treatment standpoint. So what do we know now when it comes to multiple myeloma and the biology that we didn't even know 5 years ago? I'll start with you, Paul, and then move it this way.
Well, it's an incredible question, Tania. I would say that the biology is incredibly complex, and I think that the heterogeneity of myeloma has always been very daunting to me, and I have to say very humbling, because ultimately what we're now seeing in myeloma, first of all, there was this appreciation of the tumor microenvironment, that this disease, unlike the linearity of leukemia or the relative lineage fidelity of lymphoma, myeloma was extraordinarily complex. And I always bring it back to the fact, if you think about what a normal plasma cell does, it all sort of makes sense, right? To my mind, it's the queen of immunological cancers, myeloma, because the normal plasma cell is designed to recognize an antigen, say when you're 12 years old, and then 20 years later, if you're exposed to that same pathogen, this remarkable cell will wake up and deliver you antibody literally within hours to fight that same pathogen. Now, unfortunately, when that particular cell becomes malignant, it retains that extraordinary sophistication, but at the same time, of course, it's adverse. So I think as we understand myeloma pathobiology, this complexity of interaction of tumor cell microenvironment, the immune system, the host, the patient. Every aspect of it tells me that this is an extraordinarily diverse disease. And so we must think, all hands to the pumps. It's not one approach versus another. It is an incredibly inclusive one as we tackle the disease.
Yeah, I agree. And I don't know if Joe or Mecide, you had anything to add.
I mean, all I would add to that is just taking it the step further that Paul's described beautifully, right? I mean, we almost say it was back in the day when it was described as multiple myeloma. It was called multiple because it was really identifying multiple plasmacytomas throughout the body. But its heterogeneity is perhaps greater than almost any other malignancy, and Paul's outlined that beautifully. I think we have done some groupings of some of the biology, but even with that, even just recently with our joint IMWG and IMS classification of high-risk myeloma, it's become very difficult. So to me, the take-home message from it is I have to look at every patient individually, which I should be anyway. I say all the time to my mentees, I don't treat myeloma, I treat people.
Yes. Yeah.
And so when you're looking at that person, you look at the whole of the context. Like, what do we know of the biology of their disease?
Yeah.
I sometimes use the analogy, what do we know about the horsepower of this engine? But then pragmatically, how is that horsepower being used, right? Sometimes the Prius is passing the Porsche, right? So we have people who on paper doesn't look like they have high-risk myeloma. You'd expect them to do well, or vice versa, they have very high-risk myeloma, but that Porsche is not driving fast, or the Prius is driving quickly. So it requires us to have this sort of dynamic response to patients, always putting them in their own social context, always putting them in the context of who they are and what their preferences are. And I think that's what's perhaps changed the most, is not just using, as Paul said, that sort of cookie-cutter approach of treating everybody the same way. And lastly, I think what makes it even more challenging is that because it is relatively rare, 2% of all cancers, and we estimate 75 to 80% of people being treated for myeloma in the US are treated in the community, it makes it very hard for that community oncologist who's treating the big four, breast, lung, colon, and prostate, and 30 other tumor types to enter that complexity when they are treating their patient with myeloma, which I think underscores all the more the importance of a dynamic relationship between the expert myeloma community and the general oncology community to support each other. Otherwise, I don't know how these community oncologists could do it. It's so challenging.
Yeah. And I'm going to throw it to you, but can I ask you a different question?
Yes.
So we talked about how much everything has changed. What is the biggest breakthrough over the last 12 months that you're excited about?
Well, I think for me, bringing in new therapies as quickly as possible is really critically important. We made a lot of advancements for patients, but when we look at how we have designed studies in the frontline setting, where to get a full approval, it's based on progression-free survival. And because of the innovation in the past decade, we know it takes many years to achieve that endpoint, right? So I think for me, a critical milestone in the myeloma treatment landscape was getting the FDA to recognize MRD as a surrogate endpoint and be incorporated now as a co-primary endpoint, where we can now get earlier readout based on MRD negative CR rates as a surrogate to progression-free survival. And maybe just to give some context, MRD means that you can detect at levels right now, but using next generation sequencing like clonoSEQ, you can actually detect minimal residual plasma cells that are clonal at 1 to 100,000. That's 10 to the minus 5. And now we're actually also incorporating 1 in a million. So what this now means is that we can actually bring in faster new therapies to patients. So traditionally, when you do randomized phase 3 studies, you had to enroll several hundreds of patients, right? 800,
and now we don't wait for OS, especially if you're talking about the front line. Yes.
So, enrollment takes 2 to 3 years. Yeah. Then waiting for that PFS to happen, another 5, 6, 7 years. It could take us from start to end a decade to bring new innovation in the frontline setting. So now, actually, you can validate an early readout at 9 months, 12 months based on MRD CR negative, and you can actually bring in these therapies from an accelerated approval, but still make sure that you follow for that final primary endpoint of PFS. So to me, this is going to revolutionize how we're going to bring in new therapies, not only in the frontline setting, but early relapse.
Yes.
And that's where I think we have an opportunity to potentially bring in more functional cure therapies for patients. And I think that's the gold standard we need to achieve for myeloma.
I think you've read my mind because, I'm excited to talk about MRD. Before I go into MRD, I just didn't know if any one of you wanted to answer that question as well. What exciting innovation happened in the
Well,
last 12 months?
it's a beautiful question, Tania. We need every option, and I think that's the point. It's not one particular exciting option, it's the multitude,
Yes.
because all of our patients are different. Each needs a specific tailored approach to their management. And what I'm so pleased about is that we have 20 drugs, if you take the whole field over the last two and a half decades, and in that we need all of them. And I think it's so important for our regulatory colleagues to understand that. It is not one versus the other. The important point is to understand the sophistication of the modality and its uniqueness, but then to recognize all these other therapies really matter. So if you ask me what I think are the most exciting things, I would say that the availability of treatment options that can be applied broadly.
Yes.
And we don't sort of have a Ferrari for the elite, we have an everyday vehicle for everyone. We've got that
I would
whole
like a Ferrari for everyone, though.
Well, in a sense, yes, except it's...
You get a Ferrari.
You get a Ferrari, yes.
And you get a Ferrari.
Well, I love that idea. So basically, my point is simply that what I'm really excited about, and it's very important to share with our audience, is that we have a multitude of options.
Yes.
Oral therapies, truly off-the-shelf outpatient therapies, truly off-the-shelf, not sort of make-believe. You actually can say to your patient in the clinic, we're going to do this like now or tomorrow.
Yes.
And we're going to get it for you, and it's accessible. And we don't have to wait 3 weeks for an insurance approval, 5 weeks for a bed, how many weeks for T-cell, generation. We can move straight in with therapy. So I'm particularly excited, actually, by the exciting new oral options for our patients that are right there. And I'm also by the fact that we've got such an extraordinary research community in myeloma that is really pushing from all sides to improve outcomes. So not one thing, but just a sort of collective view.
Yeah, and I think the pipeline of medicines that's coming out, to your
Exactly,
point, even
exactly.
that whole thing with allowing MRD as a surrogate endpoint is going to allow us to move a lot faster, as you mentioned.
Yeah.
What about you, Joe?
I absolutely agree with my dear friends here. I think what I would add to it was, and it wasn't really even about the one study as much as the concept that we are now, for genuinely the first time, and not just in this sort of distant way, starting to having to define what cure is. And this was a product of, the long-term follow-up of the CARTITUDE-1 CAR-T trial, where 1/3 of those patients, and granted we're talking about 32 patients, but 1/3 of the patients on the trial, after a single infusion, 5 years later, are still completely disease-free.
Yes,
For those
agree.
in whom we tested MRD, they were MRD negative.
Before we continue, let's quickly discuss what CAR-T therapy is. CAR-T cell therapy is a type of adoptive cell immunotherapy that uses a patient's own T cells, their natural defenders, and re-engineers them to recognize and destroy myeloma cells. So we start by collecting the patient's T cells, and in the lab, they're genetically modified to express a chimeric antigen receptor, or CAR, usually designed to target proteins on myeloma cells. And after the modified cells are expanded to create millions of copies, the patient receives a brief lymphodepleting chemotherapy to make room in the immune system. Then the CAR T cells are infused back. Once inside, they act like guided missiles, actively seeking out and binding to myeloma cells through that CAR antigen interaction, releasing potent cytotoxic signals that trigger tumor death. So basically, personalized therapy, reprogramming the patient's own immune system to fight their cancer. Okay, now back to the discussion.
So if we go back to when all of us went to med school and survival was typically 1 to 2 years, there was always a small fraction of patients who with minimal intervention had a very long-term benefit. That fraction has gone from a few percent to now hopefully this study showing to us that we can get at least over 30%, and of course the hope for more. And it's caused us as the myeloma geeks of the world to say, okay, we need to actually define cure.
Yes.
I like to define cure the way someone on the street would define cure. I guess you get treated for a period of time and then you don't have to think about disease anymore. We may not be able to achieve that in the majority, but that fraction is growing. So I think it's given us, without overusing the analogy, that light at the end of the tunnel. Like, we are seeing that there is a bigger gap at the end of that tunnel. We can get more patients to that. And lastly, what was lovely about it, of course, is that these are patients that had a one-time intervention. This idea of ongoing therapy for everybody forever is challenging.
Yes.
And I think the goal is to treat for whatever defined period of time we think is appropriate. Some may require longer or lesser based on the biology that we've just described. But for us to be able to take a patient and say, I'm going to give you this one intervention, and then you're literally going to be off therapy on my favorite drug, nothing. Right? Every insurance company covers it. Patients are always 100% adherent. But the idea is that long term they go that, and that's not make light of something so important. I think of a lot of my patients and whom we've been able to either take off treatment or give a one-time treatment like this, or treat them for a while and stop a bispecific, whatever it might be. It is incredibly empowering for them to be off treatment and to enjoy their life, because that's really ultimately the goal, right? It's not just about the time, it's the quality of the time that they have.
And I'd like to build on that, if I may, Tania, because I think it's a great point Joe makes, but I also think it's very important to note that we also realize that for a substantial number of our patients, that one and done type approach will not apply, be it even in the most successful CAR T setting, where the results are nothing short of miraculous. What we're realizing, though, is that there is this substantial number of patients who need a maintenance approach to maintain remission. What I'm excited about is we now have the toolbox with some very exciting new oral agents that are so exquisitely well tolerated and don't have late complications that might have, to some extent limited our previous options. Now we have that toolbox available too. So to Joe's point, I couldn't agree more. One and done's wonderful, and NADA, I love that. NADA is my favorite drug. But I will say, for speaking to the majority of practice, continuous therapy is part of our management strategy in myeloma, well established. But what's so exciting is those continuous therapies continue to improve, and their tolerability profiles are such that, my patients are fine. Yeah, hey, look, I take my Crestor, I'll take my oral agent to keep my myeloma away. Fair enough. And as long as we can give them that kind of quality of life and that kind of ease of use, then I think we're really making headway.
Yeah. So let's start there. Mecide talked about MRD. At what point of MRD negativity in a patient who's on maintenance therapy, when do you say enough is enough and we can actually pull back?
It's a great question. I think we're realizing there are very important aspects to MRD. One, it can be sometimes somewhat unstable. In certain sense, interesting enough, in the CAR T setting and the relapse refractory setting, I think we've recognized that's a particularly unstable signal, that it has to be sustained. I think post-induction remission therapy, whichever stripe, followed by consolidation and then maintenance, there is a sustainability in that setting that's more reliable. And so if you have MRD negativity for 2 to 3 years on therapy, there is a lot of thought in the field that that's a junction point at which we should try and understand if we withdraw treatment or just complete it and then de-escalate and observe, is that an opportunity? And there are trials looking at that. I will say I have some caution about that approach outside of clinical trials, because I do think I am forever humbled by myeloma, and I'm humbled by that pathobiology we talked about earlier, its ability to wake up 20 years later and come back. And if there's a strategy that allows us to safely do that and achieve functional cure, wonderful. But I think we need to do that in the context of trials, and I think we need to make sure that we're being driven by information that guides us, because, again, if the maintenance therapies were too toxic, there would be a much different discussion. But the point is, with that in mind, we need to be then very thoughtful about de-escalation, and in particular, completing therapy.
And in a lot of your talks, you talk about hitting hard early. Right, we have now, and again, the treatment paradigm to me is getting really complicated. We started with triplets, now we have quads, we have CAR T, we have bispecifics, ADCs, right? But how do we sequence? How should we start? And if you look even beyond just hematology, think through solid tumors. A lot of people say breast cancer, start hard, hit hard. Colorectal cancer, start hard, hit hard. And then the question is, if you do that, do you end up building resistance down the line? Can you tell us your thoughts on
I wish I had the answer.
therapy?
Yeah, I always say, if you want to host a myeloma meeting and have people come, entitle the meeting "The Optimal Sequencing of
That's the
Therapies in
discussion
Myeloma,"
now.
because it's a bit of that pink unicorn that everybody is seeking after and nobody fully knows. But I do think there are some key lessons that we have learned. I think the notion of hitting hard, hitting early, as you described beautifully, has a lot of biological validity in myeloma, and maybe even more so than many of these solid tumors, because of the complexity of the plasma cell that Paul described earlier, that this disease has the capacity to overcome treatment, to become resistant, to mutate, to change, so much more than almost any other malignancy. So I think we have learned that in general, more is better up front. And paradoxically, I think when we can use more, we can often shorten the total time of therapy, as opposed to constantly giving very low dose, smaller treatments. Meaning, how did we overcome HIV? Not that we fully overcome it to a certain degree, but it was not because we had that one signature drug. It was the right combination, right? And in a disease that has a complex biology, and until we can truly identify the biology in every one patient and truly match it to the treatment, it makes sense on the whole to treat with a broader group of drugs together. So I think right now, the most important principle is that we use these combination therapies up front. We're trying to minimize their toxicity. We're trying to optimize how long do we need a transplant, do we not need a transplant. As I said earlier, I wouldn't be investing in transplant stock right now. I'm not sure that that's going to remain. But then we get hopefully the vast majority of patients to a lengthy remission. I mean, the prediction now after a quadruplet and transplant is that these patients are probably going to go over 10 years on average before their first remission. Then the sequencing thereafter really depends on the scenario. That person who goes 10, 12 years is viewed at very differently than the person who relapses one year after, right?
Yeah.
And then, of course, we have multiple other lessons. I won't bore everybody about all the other sequencing lessons, but a couple of highlights include: don't save your best for last, right? I always say it's great for a Hallmark movie, not good for myeloma, right? Because the disease does become more aggressive. So as we've seen things like CAR T and bispecifics prove themselves in very heavily relapsed disease, we're going to be bringing them earlier on. Also, to listen to the patient, right? This concept of shared decision making and patient preference isn't just a let's hold hands and sing Kumbaya moment. Like, it really is critical. With all the choice we have now, jokingly, the Cheesecake Factory menu of choices, we want to sit down with our patients and say, is this a one and done approach for you? Would you rather take that lower dose therapy and have it continuously? Does this fit with your lifestyle? Now that we have choice, until we know what that ideal sequence is, I think it's very reasonable to make sure we include the patient. And then lastly, that the biology guides some of it. We know that going from one exact target to another, from BCMA to BCMA, can be done, but does it have to be done? Can we switch targets? Can we switch approaches? I think those are some of the overarching principles to sequencing. I'm just encouraged that for most of these therapies, they become in play. Like, when we started treating with CAR T, bispecifics, and antibody drug conjugates, we had this feeling like you've got to choose one of those three bridges. And once you choose it, the other two are burnt.
And now
that's not the case anymore.
Yes.
Absolutely.
Yes. Yeah.
Absolutely.
Yeah, maybe just to also add to the fact that, later lines, there's such a high unmet need. And I think with us seeing such a wonderful opportunity to deepen the remissions and the durability in the newly diagnosed setting, early relapse, it drives innovation to really think differently and move new therapies, modalities into the late relapse.
Yes.
Such as now, there are like trispecifics that are being explored, right? So you're going from bispecific to trispecific and different targets to address the complexity as the myeloma goes through the evolution of the clonality. And so I think it's actually a very exciting time to not reserve your best drugs to later, because it's going to have to drive the science and us to do better to innovate in later lines, so that then we can move those into earlier lines to continue to evolve the innovation and have the most impact.
And I would echo, beautifully put by the chair, and of course Joe's comments are spot on. I would just say a couple of more points though. I think as we think of moving strategies earlier, we have to bear in mind safety.
Yes.
Because I think at the end of the day, when our modalities are exquisitely safe and they're not long or late or short-term toxicities that could compromise outcome, that becomes very germane. And I think for my patients, as we look at the landscape and think strategically, I so agree the quadruplet strategy, which is, you know, and the patients look at me and say, well, is it chemotherapy? I always say, no, it isn't. It is biologic treatment, and it's actually all immune-based, actually. Everyone says we're living in the era of immune therapy. Well, actually, if you think about it, myeloma therapy has always been immune-based. From the very start with high-dose melphalan stem cell support, it was actually immune treatment. So we've always been in immunotherapy. It's just that immunotherapy's gotten so much better. And I think at the end of the day, with quads being so successful, if you think of this, Tania, a quadruplet can gain for your patient without transplantation necessarily 8, 9, 10 years of survivorship and progression-free survivorship. And then when they relapse, and hopefully they don't, but if they do, then we look at options that will buy 3, 4, 5 years. And suddenly you're looking at a landscape that's 15, 20 years out. And for my patients, you know, median age 75, 80, this is suddenly, you know, this is a functional cure, essentially. So I think the strategic considerations become very real to bear in mind what you do early matters later, sure, but also we must make sure the early platforms we use have exquisite safety.
Can I just emphasize one point that Paul made? If there's a 3-word phrase that I use in my clinic repeatedly, primarily in the research context when residents and fellows and others are working with me, I say it repeatedly, I say safety is paramount. And now that myeloma patients are living longer, it's not just as simple to say, okay, they're having a greater quantity of life, now let's also focus on quality of life, which is true. I mean, I think of the beautiful story you shared. I almost got choked up listening to you, Paul, you know, about your patient who you brought out to see the sunshine. I mean, I think one of the reasons why I went into myeloma is I would go to the myeloma clinic and I'd look at the waiting room and everybody was hunched over, everybody was in pain. And I thought, these are the people that I want to help. I want to find a way to improve it. So it's not just about quality, but that degree of safety is fundamental. And we've seen this already with the classes of drugs we have that we develop. We prove the principle with whichever class, a proteasome inhibitor, an immunomodulatory drug, a monoclonal antibody, and then we say, okay, we prove the principle. Can we leverage that same mechanism, but do it safer? Can we go from bispecifics to trispecifics? Can we have a dually targeted CAR T that may be able to produce it more safely and potentially even more effectively? And that is what really excites me as well, is that the standard is getting higher and higher. I've said, you know, we've gone from sort of economy class to economy plus to business class to first class. I don't know if we have to sit in the cockpit, but we're trying to move things forward. But that safety is so fundamental. And then if I can add even one dimension to that, even more convenient as well. You know, can we do it with less hospitalization, with shorter time on treatment, with a more rapidly delivered product, with less of the other sort of known side effects? Because I think as myeloma doctors for years, we and as transplanters, we just sort of say, well, you know, you're going to go through this, you're going to lose your hair, you're going to be sick, you're going to have a fever. We kind of just accept those. I want to not accept those anymore. I want us to be able to say that we can deliver this as much as possible. Even our solid tumor colleagues are giving oral drugs for treatments that they used to give heavy-powered chemo. I think we can do the same.
So now you said, we talked about CAR T, can we make it safer? And the question I have for you is, do you think there's going to be a time where CAR T fully replaces transplant and transplant just becomes a salvage?
You know, it's so interesting. I think that's a great question. I would echo Joe's point. I really believe this now, less pain, more gain. I really believe that. I think that's a fun, I love some of your comments, and Joe, I think I love the less pain, more gain one, because as originally as a high-dose therapist myself, you know, there was always this axiom that, you know, more pain, more gain, and I think that's absolutely incorrect. Pathobiologically, it's actually not correct either. But the fact is, going forward, will CAR T replace transplantation? I think what I would say more broadly is that immune therapies, as we're developing, be they CAR T, be they bispecifics, be they trispecifics, will increasingly replace the need for high-dose melphalan with autologous stem cell support. And moreover, we will have other ways of delivering the benefit of high-dose melphalan without the side effects and the long-term consequences that can be so challenging, be it increased mutational burden at relapse, or unfortunately, a continuous increase in the risk of second cancers, and in particular, myeloid malignancies. So this is something we must get away from, because unfortunately, I have lost too many patients to secondary AML. I know Joe has too. You know, we realize it's not a huge problem, but it's a problem that isn't going away. And what's even more important, as patients live longer, it actually becomes more of an issue. And if you look at CAR T, for example, you know, there is a real signal for MDS underlying CAR T, and it seems to be driven if you've had prior melphalan. So then you kind of need to think about, well, sequencing there obviously matters. So to answer your question, Tania, I would say we'll use transplantation more selectively and less. Having said that, we will replace it with more rational immune therapies that will be tailored to the patient's need.
I just want to add on what you said, Paul, that even today in clinical practice, we know that patient may opt to defer the transplant based on the MRD status, right? And what we are seeing, the trend is that at the time that they relapse in the second line setting, they're actually then moving forward with the CAR T. So, and I think we're looking forward to see the pivotal data, CARTITUDE-5 in non-transplant eligible patient, CARTITUDE-6 and transplant eligible. I think those studies are going to really help guide us. And maybe there is a subset of patients, as you alluded to, that will still benefit from high-dose melphalan. And even if we see a benefit of CAR T in the frontline setting, we still want optionality for patients because we know that patients may want to still defer it to later line. And
Mecide, I would reiterate the point though, because I think we all recognize as myeloma physicians, there is really a value to the alkylator warhead. We realize that. I mean, one mustn't diminish that. I mean, it's improved survival over decades. The important point is, can we do it more safely? And I would really say we obviously use cyclophosphamide as a backbone drug in many situations. And I also am really an enthusiast for this targeted approach of peptide drug conjugate delivery, because it gives you the value. It's a Trojan horse, to paraphrase Pete Voorhees, because it gets into the tumor cell and also opens the gate to bring more prodrug in to then convert it into being more active. But very importantly, as we look at the specter of things like extramedullary disease and this stemness in myeloma that resists immune therapy, unlike leukemia where it may be a leukemic stem cell, I think stemness in myeloma is more complex. It's a sort of functional unit. And targeting stemness is tough for our immune treatments. And the hallmark of that was allotransplant. When allotransplant failed our patients, extramedullary disease was typically what we saw. So when you look at EMD, that to me is a surrogate for for therapeutic failure in a patient that we need to think about. And therein, I think, lies the value of targeting stemness with drugs such as peptide drug conjugates, which are a smart way of delivering your melphalan warhead, and at the same time bringing other agents. And I'm obviously particularly encouraged by the efficacy of drugs like mezigdomide in the setting of getting into EMD. And I can see a way forward for combination strategies there that could really matter. So I think that's important to share, because you know, this complexity of natural history of disease, the clonal tiding, Mecide, that you alluded to before, we have to have options for our patients at each part of this journey. So anyway, I hope that helps in the context of our conversation.
And I think the other way to put it is we're not just going to wake up one morning and transplants can be replaced by CAR T.
Exactly.
You know, I think in any disease that we're not curing at least a significant fraction of patients, we want more tools in the toolbox.
Exactly.
And then if I can put my IMF hat on for a moment as the Chief Medical Officer of the International Myeloma Foundation, the I in IMF is international.
Right.
And that's not to say that we have lesser therapies around the world. It's really a question of ensuring we have access
to it,
I am a little bit excited about some of the concepts around allo CAR T, where you could potentially do an off-the-shelf CAR T. We're actually just looking at a trial literally this week of introducing that. But I think we also have to have that perspective. We've looked at it domestically here in the U.S. to say there are areas where people have reduced access, whether it's transplant or CAR T. I mean, for years we've known if you grew up in rural Mississippi or Alabama, it's going to be much more challenging to get these therapies. And so I think this idea of having as many tools in the toolbox so they can be applied is very important, both biologically and pragmatically. And of course, I can't help, and I don't want us necessarily to segue here, Tania, but you know, I can't help but note that myeloma has been a disease of tremendous disparity.
Yeah.
And arguably the greatest disparity within the African American community. A black man or black woman diagnosed today with myeloma is expected to live half as long as the same aged white man and white woman. Now, some of that is myeloma mortality, some of that is other health mortality. But the point being is that there's been that huge disparity, and all this excitement about new therapies is very appropriate. But we want to, and we must, I should say, we must be able to approach it in such a way to ensure that it's equitable to all.
I'll follow your lead because, to your point, twice as likely to die from it, twice as likely to be diagnosed. However, as you know, the data shows that when you give them equal access, they're not twice as likely to die, but live as long as their white counterpart.
If not longer.
If not longer. And that's what I'm saying,
Absolutely.
when you look at the VA study, even longer. Let's talk about this. Is it biology in terms of let's start with why are they twice as likely to be diagnosed? Is it biology? Is it environment? Curious to get your thoughts. Maybe Paul looks like he's ready, but
it's such an incredibly important point. I want to just touch on, before we just dive into this about disparity, the value of access and what that means. Because I'll just share with you a story. Ixazomib was just approved in Indonesia. Indonesia is a huge country with literally it's the largest Islamic population outside of the Middle East, to give you an idea of that enormity. Ixazomib is a game changer there because it's an oral proteasome inhibitor that can be delivered to the island peoples easily and require minimal exposure to clinic. And I'm thinking of the value of oral therapies. And I was just struck by it, and how there was a launch meeting, and literally all of these physicians from all over Indonesia showed up because this was a way of getting proteasome inhibition in a pill for the people who live in disparate islands all over this whole enormous country. And it just sort of struck me to your point about IMF, and good for you, Joe, because I think that's essentially a key point. So pivoting back to the disparities in healthcare in the US and these challenges in our African American community that exist, I think Tania, your point's so well taken. Let's focus first and foremost on biology, because I think you're absolutely right that there's pathobiology that matters. And my own observations there on our own teamwork in that space has been driven by observations from the determination trial, where we were so fortunate because we had free drugs. We were able to leverage access with that trial, which we launched in 2010. And as a result, and we had a very deliberate strategy, our team. I was so lucky to have co-investigators of African American heritage. We worked very hard. There was one particular colleague of mine, Tondre, who moved from Mississippi to South Carolina, and I moved the study with him because we had this absolutely determined view that we must improve access for African Americans. And bingo, we had almost 20% participation. And it was a privilege to have Joe as a discussant at the plenary session at ASCO.
Sorry, just for our listeners, that's rare, because when you look at African Americans in clinical trial, we're looking at, I mean, you're looking, what, 3 to 5%, if even, right? So to have 20% is huge. So
Right, well, thank you, Tania. But it was a very deliberate strategy, and it was sort of a boots on the ground strategy, as in physician colleagues who we knew would be attractive to the African American community because they were African American. We had a great exposure in the southeast, deliberately on that same basis, and we moved the study literally with colleagues to make sure that sort of continuity of access would be there. In any event, what we found was, to our surprise, where you might think, for example, transplant has to be offered to every African American patient, and if you don't, you're doing them a disservice. Well, sure, of course they should have the option, but what we learned in determination, in an absolutely, controlled environment where access was not the issue, socioeconomic status was not the issue. What was the issue was biology. We saw remarkably that African American patients appeared, whilst they did benefit from transplant, actually they even did better if it was kept in reserve. And this to us was a complete surprise. And I'll never forget Joe discussing it at ASCO, because he presented a case of his African American executive who wanted to defer transplant for career reasons and family reasons. And why it sort of started to really resonate is we took a deeper dive on that, Tania, and we started to test a hypothesis around what we call Duffy null. Duffy null is a red cell phenotype that's been selected for over the centuries, over the millennia, because it protects against the consequences of falciparum malaria. So in equatorial countries, it's obviously a huge survival advantage. So that means Africa. It means also by virtue of migration, Latin America. It also means, of course, the Middle East. And Duffy null occurs in up to 65% of people of African heritage and the Middle Eastern heritage. What we found so interestingly is that this treatment benefit of keeping transplant in reserve magnified in those patients who are Duffy null versus non-null. And that was irrespective of whatever ethnicity they declared, be it African or otherwise. This was remarkable, and whilst it's a preliminary finding, we're really hoping to better understand this because it may mean
I mean, maybe you can explain why you believe.
Well, I believe that
Yeah.
because I think critically, Duffy null is about how the body handles inflammation. And if you take away the red cell sink in the body to handle inflammatory consequences, unfortunately, inflammatory conditions are going to be much more challenging for you. This may explain a lot, actually, in terms of diabetes, obesity, cardiovascular disease, which all have an inflammatory basis. And if you are Duffy null, you're less able to handle inflammation. Your inflammasome is different. This may explain, in part at least, why myeloma is more of an issue. In fact, de novo, it may be part of the etiology of why myeloma is more common in people of African and Middle Eastern heritage. 2, tolerance of certain treatments may be an issue. And in fact, consequences of treatments may be an issue. So to your point, Joe, less pain, more gain. If we can make treatments more refined and more tailored, RVD performed very well in our African American patients in determination. Our African American women of higher BMI, the median PFS has not been reached.
Really?
It's not been reached on our analysis. So it tells you that if you've got someone who may be hurt by too much inflammation, be it transplant related or otherwise, use your toolbox, but a quad would be perhaps ideal because you don't inflame. You actually offer a quadruplet therapy. And as we talked about earlier, for our Duffy null patients, for treatment effect, keeping transplant in reserve, we saw a hazard ratio of 0.21, which is striking.
Which is
So because of that, I mean, we have to be very careful because these are hypothesis-generating subgroup analyses. So just to be very, very correct about that. But this is a remarkable observation because it ran completely counter to what we'd expect. And so I think as we think forward, we want to think creatively about how we tailor therapy. And so I argue that the quadruplets are a wonderful advance. Keeping stem cell transplantation in reserve for patients, especially if they're Duffy null, may be a very wise move. And at the same time, we obviously need to prove this with further studies and be sure of our ground. But I'll share an example. I have a wonderful gentleman who's a basketball coach. He literally ended up with very advanced disease, put him in bed. He could not get out of bed. He had extensive myeloma. He's of African American heritage. We treated him with quadruplet therapy. He's had a CR and he's MRD negative and he's Duffy null. And we had this chat and he looked at me and said, Paul, we're keeping that transplant in reserve, brother. And guess what? He's coaching and he looks $1 million. And he's done really well.
So what I would add to that is, so first of all, Paul, thank you for doing this work because I think it is important. And for a long time, people used to just say, oh, well, I ran a classical heme, as we call it, or benign heme clinic for years when I was in Toronto. It was such a multicultural city. And we would just say, oh, well, we just expect African-American individuals to maybe have a slightly lower white count. We hadn't discovered Duffy null yet. So I think doing the work in that area is really important. Answering your initial question, is it biological? Is it environmental? Is it social?
Yes.
It's like I say, my favorite answer to any question is all of the above, right? And that's not to make light of it, but it really is all of the above. There's no doubt that individuals like myself of African descent, we see a higher incidence. Often it's not been measured in these countries because if you don't take a temperature, the patient won't have a fever, right? We just haven't looked for it.
Yeah.
In the countries that we have done screening studies in Uganda and other places in Africa, we have seen this higher incidence. But then we also see here in the US, it's not just about the higher incidence, it's about the delayed diagnosis and the reduced access to care, along with all the other social determinants of health. Now, you know me, and we could get talking about this
Let's do
all
it. Yes.
day long,
Yes.
but just super briefly, I think we have to understand that mortality difference is not just a function of do they have access to transplant or CAR T or, the big 4 Ts that I call triplets, transplants, trials, and CAR T that have all propelled myeloma forward. A lot of it is about the basics of life and housing insecurity, being able to have access to primary care, being able to access to simple food, right? I mean, I did a bit of a study. I live in Arizona and it was involved with my work at Arizona State University within undocumented women with breast cancer that they were had developed this amazing program to provide treatment for them for their breast cancer, and even provided transportation. But we found out that some of these women were not coming to their treatments. And we found out later, they said, well, if I get treated and I'm even just a little bit nauseous, nobody in my family eats tonight because I'm responsible for making dinner. Things that we as clinicians maybe not always think of. Your patient's not going to listen to you if they're hungry, right? So those social determinants of health, not that we can fix those overnight. They've taken, sadly, centuries to create, but we need to address them. But then also on the myeloma-specific side, the delayed diagnosis, to me, is a particular point. And Paul even raised it when he commented about other syndromes and other scenarios. There is still a bit of a dotted line connection between myeloma and obesity, as with most cancers and obesity we don't fully understand. The other stronger dotted line, I would argue, is with diabetes. How often have we seen our patients, with myeloma say, well, I had diabetes, and for several months, my doctor said my anemia, my renal insufficiency, my proteinuria, my neuropathy was all because of the diabetes. I'm not saying the diabetes causes myeloma, but it's the mimic, and it contributes to the delayed diagnosis. So as we look to solve this, we have to look and understand the biology more,
Right.
address the social determinants of health, consider how we make the diagnosis of myeloma with our primary care providers, and then indeed look at the access therapy. So I think the access to therapy is a huge issue, but it's preceded by these three others that I believe in. And I actually think we have been making progress. When I quote earlier that a Black man or Black woman diagnosed with myeloma is expected to live half as long, those numbers are improving. And they're improving because of this multifold strategy.
I would argue as well. I think you're absolutely right, Joe. I think I would argue because to the point we made earlier, access to triplet, quadruplet therapy is improving. And so, it's very interesting to me that those outcomes are better because, in a sense, if transplant was the winner, why hadn't we seen it sooner? Because I know, for example, in centers like Emory, where they've made a very deliberate effort and done full kudos to them, huge applause to them for doing it, and Arkansas as well. They've deliberately tried to do this, but the needle hasn't really moved. What has really moved has been the advent of biological therapies and their delivery in the community, as well as, of course, the fact there's maybe a pathobiological reason why they'd actually do even better. So it's a great observation, Joe.
And I know I could talk about it all day long. We need to move on, so I will stop after this. But the last thing I'll say is that, it really does take a multimodality approach to this, and it takes everybody, our partners in industry, the lay public. I have the privilege of working even with some of our friends in Hollywood to raise awareness around this. I mean, I believe that this notion of you only think about your healthcare provider or your local hospital when you get sick has to be reversed, that we as the healthcare community are going out into the community, building trust, building connection, emphasizing preventive measures of health so that when people do need that help. I'm privileged to do these programs in churches and fraternities and sororities and community centers through the IMF. We call the Empower program.
And please, all of you who are listening, you're going to see this. I see it on LinkedIn. I see it throughout social media. Attend. And
part of the goal of going to these churches and these places to say, not to frighten people about myeloma, but here are the basics of myeloma, because we know there's no hallmark sign or symptom, the fatigue, the bone pain, the anemia. But if people understand more of the impact it has primarily within the African American and Latino American populations, then they can do more to seek the resources and the help that they need.
That's just the beginning. Stay tuned for part 2, where our experts go even deeper into what's shaping the future of multiple myeloma treatment.